Welcome to the Chapter 2, Section 4 podcast on chemical reactions and enzymes. And let's get started. Chemical reactions, the process that changes one set of chemicals into another set of chemicals. If you look below, I show a chemical reaction between hydrogen and oxygen. And those chemicals, the hydrogen and the oxygen chemicals, become water. Some chemical reactions occur slowly. Here's an example of rust. The chain there is showing rust after many years. Iron and oxygen combine together to form iron oxide, which we also call rust. And other reactions occur quickly, like this burning of hydrogen inside the Hindenburg. Now, when a chemical reaction, we look at it as what goes in and what comes out. The components that enter the reaction are the reactants. The compounds that come out of the reaction are known as the products, what's produced. What happens to chemical bonds during chemical reactions? The chemical reactions always involve changes in the chemical bonds that join atoms and compounds. And as you know, we talked about the electrons being what cause the chemical bonds. Energy in reactions. Energy can be either released or absorbed when chemical bonds are formed or are broken. And it always involves breaking. They always involve changes in energy, no matter what happens in a reaction. You can see the chemical reaction occurring in this fireplace is releasing energy in the form of heat and in light. How do energy changes affect whether a chemical reaction will occur? The chemical reactions, if they release energy, may occur spontaneously, and chemical reactions that absorb energy will not occur without an initial source of energy. So let's look at hydrogen and oxygen combining together to form water, and it's an energy releasing reaction, which means it's going to give off heat as it occurs. And if we reverse this reaction, we would have to add energy into it, meaning if we were to try to take water and split it into hydrogen and oxygen, we'd have to add energy into it. And this is a science fair project often seen is using electricity to separate water into its parts of hydrogen gas and oxygen gas. In order to for organisms to stay alive, they need to do reactions to carry out energy. In this case, we're looking at photosynthesis, where the plant is actually taking in light energy from the sun. And so in order for organisms to carry out chemical reactions, they're going to have some source of energy. And once again, plants get their energy from the sun, and also animals need to get their energy from eating plants or other animals. Activation energy. So in order to release, if you remember correctly, we said that chemical reactions that release energy don't always occur spontaneously. You need energy to get the reaction started, and that's called the activation energy. If you look at the two charts below, you can see that the energy absorbing reaction needs a small amount of activation energy, and then you end up the products have more energy than the reactants do at the end. And if you look at the second chart, where it's an energy releasing reaction, you see that the products have less energy than the reactants, but still they need the activation energy in order to get that reaction started. So activation energy is a factor in whether the overall chemical reaction releases energy or absorbs energy. Enzymes. In order for reactions to occur, to make life possible, often you need these reactions to occur quickly, and some reactions don't occur quickly without the help of an enzyme. And enzymes are a form of a catalyst. Catalyst is a substance that speeds up the rate of a chemical reaction. They work by lowering the reaction's activation energy. If you look at the chart below, you can see that the blue line shows the energy required for a reaction to occur. The red line shows what the energy is reduced if you add the catalyst to it. Why are enzymes important to living things? Enzymes are proteins that act as biological catalysts. They speed up the chemical reactions that take place in cells. And they do this by lowering the activation energy. And you can see on this chart here if you lower the activation energy with it, by looking at the red line, you can see that without the activation energy is lowered, the reaction is going to occur quicker. Enzymes are very specific. They generally only work on one chemical reaction. If you look at that picture over there, that's a picture of an enzyme, and that enzyme is very complex in its shape. And we often give the name to the enzyme from what reaction it catalyzes. DNA polymerase is an example. What it does is it helps catalyze the polymerization of deoxyribonucleotides into a DNA strand. In other words, it helps construct DNA from the deoxyribonucleotides. For a chemical reaction to take place, the reactions must collide with enough energy so that existing bonds will be broken and new bonds will be formed. And if they don't have enough energy, no reaction will take place. We'll refer back to this later on in the year. So you saw that complex picture of that ribbon looking enzyme. Here's a more general model of it, where we can talk about the enzyme substrate complex. What the enzymes do is provide a specific shaped site so that the reactants can be brought together to react, and that reduces the energy needed for the reaction. The reactants of these reactions are known as the substrates. So if you look at that gray blob is the substrate. The enzyme, which you can see is shaped very specifically for that substrate, joins together and creates the products. And so this is just an example of glucose and ATP binding to the enzyme hexokinase, forming an enzyme substrate complex. And the shape of the enzyme is so precise that it fits so well the active site that it's often compared to a lock and key. If the enzyme is not going to work in other reactions because it's basically constructed in such a way so it will only fit on a certain active site. There would only be one key to each lock. And the enzyme and substrates remain bound together until the reaction is done and the substrates are converted into the products. So you can see this is a cycle here where we have a enzyme, which is that peach colored blob in the center. The substrates would be glucose and ATP. The substrates bind to the enzyme in letter A, and then they're converted into the product, which would be in this case ADP and a molecule of glucose, and then they're released, and then the enzyme is free to start the process over again. It's not used up in the reaction, it's reused again and again. And enzymes can be affected by any variable that influences the chemical reaction, such as pH. They work at best at certain pH values. You would think that most reactions would work very close to pH of 7, although in your stomach you'd probably think they would work at a much lower pH. And also enzymes are affected by changes in temperature, and as it occurs, most enzymes work very well at human body temperature, which is about 98 degrees Fahrenheit. Cells can regulate the activity of the enzymes. They often have a key that turns the enzyme on and off at critical stages during the cell. And enzymes play roles in chemical pathways, making materials that the cells need, which we've seen already, releasing energy, which we just saw, and also in transferring information, which we'll see more when we get to RNA and DNA and more about the cells.